Hello world, it's John Pinto, your roving realtor, bon vivant video blogger, podcaster, here with Matt Tryon, nutritionist, and we're going to talk about microbiome research. And I know when I went on to YouTube, everything was at least 2 or 3 years old, and everything is 90 minutes to 2 hours coming out of Stanford or Langone School of Medicine. We're going to take 4.5 minutes here, and Matt is going to tell us all about microbiome research. Please enlighten us.
Well, there's a lot of research that's going on at universities and other institutions looking at the diversity and the impact of the microbiome as it's related to specific diseases and things that, in general, things that we do in our everyday life that impact the microbiome. So a lot of these big studies use poop samples to look at our specific microbiome. But what we're moving in towards now is a new phase of microbiome research. And we're using something called shotgun sequencing, where we get to look at the microbiome from a species level. And that gives us like a lot better resolution on what is in our gut, how it's responding, and specific species that play a big significant role in how our body's working. This is a little different than it used to be a few years ago, where we're using a different method, where we didn't have the ability to look at the microbiome at a species level. It gave us a little bit of an idea of sort of the phylum and the genus of specific bacteria, but not really the specific species involved. And that's important because there are multiple species in these phylums and genus bacteria. And they all kind of respond differently to different things, and they all communicate with each other in different ways. So having a resolution on a species level is extremely important. And now what's nice is that there's commercial kits that are available for home use. So you can actually look at what your microbiome sample is on a species level using the shotgun sequencing.
Oh, okay. So enlighten me on the shotgun sequencing. I'm not quite sure you understand what that is.
So shotgun sequencing is sort of the new way of identifying species of bacteria in the gut. And as I was saying a little bit earlier, we were using an older version of how we were looking at the microbiome. And that was called a 16S, where they took a specific gene and paired it and matched it with other bacteria that have this genome sequence, 16S. How do you say it? Like a
sample?
Didn't give us the resolution that we were looking for. So we couldn't tell what the specific species were in our gut. Shotgun sequencing is a different method that allows us to look at bacteria on a species level, which is important.
Oh, good. And are there any particular species that we're trying to propagate versus those that we're trying to avoid?
There's a number of great species that seem to help. And as an example, Akkermansia muciniphila is a bacteria that is useful and seems to be low in people who have diabetes and obesity. So one of the things that we try to target is using oligosaccharides or supplements that can help, probiotics, prebiotics, that can help raise the level of that particular species. And that actually seems to help people who have diabetes control their metabolism a little bit better and control their glucose responses to food a lot better.
Well, this sounds very encouraging because you could take a more scientific, less random approach to your physical well-being and make a personal menu for yourself that's going to be customized to your own microbiome. So with that, we'll end this video, and we'll see you on our next episode. Bye for now.